Okay, so this is a chapter on tissues. Well, we have about trillions of cells in our body, and there's about 200 different cell types. And there's 4 categories of tissues that those cells make up. There's epithelial, there's connective tissue, there's nervous tissue, and there's muscular tissue. And we've had a chance to study these a bit in the lab, looking at slides and so on. Quick review of some other terminology. An organ, which is a structure with discrete boundaries that is composed of at least 2 tissue types. And then histology is going to be it's a microscopic anatomy. It's a study of tissues and how they are arranged into organs. So we're going to start with the first section, and these are the objectives right here. We'll look at them one at a time. So our first objective is to name 4 primary classes into which all adult tissues are classified. So let's first define tissue, which is a group of similar cells and cell products working together to perform a specific role in an organ. The 4 primary tissues are epithelial, connective, nervous tissue, and the way they differ are in the types and functions of their cells. They differ in the characteristics of the matrix, or the extracellular material, and they differ in the relative amount of space occupied by the cells versus the matrix. In some tissues, it's mostly made of cells, and others it's mostly matrix with some cells here and there. What is the matrix? So the matrix is an extracellular material, and it's going to be composed of fibrous proteins and a clear gel-like ground substance. It's also known as tissue fluid or extracellular fluid, interstitial fluid, or tissue gel. Okay, so second objective is to name 3 embryonic germ layers and some adult tissues derived from them. A fertilized egg, the zygote, is going to become an embryo with layers. And the layers during their development are going to be called germ layers. Those germ layers include the ectoderm. Ecto means outer. And this ectoderm is actually going to give rise to the epidermis and the nervous system. Then there's the endoderm, and endo means inner. These are going to give rise to mucous membrane lining of the digestive and respiratory tracts, to digestive glands, and some other things. And then there's the mesoderm or mesoderm, and meso means middle. And this is going to become the gelatinous tissue called mesenchyme. Mesenchyme is wispy collagen fibers and fibroblasts in a gel matrix, and also gives rise to muscle, bone, and blood. Our third objective is to visualize the 3-dimensional shape of a structure from 2 dimensions in a tissue section. So here, when we prepare a tissue to look at under the microscope, we take pieces of it or sections, and we look at it from one angle, and it gives us a 2-dimensional. So how do we go from a 3-dimensional structure to understand what it is we're looking at, which will appear as 2-dimensional? So we have to know how they're prepared. So when we do prepare a specimen to look at of a tissue, we have to preserve, slice and section the tissues. In order to preserve it, we're going to use a fixative, which is going to prevent decay. One common fixative is formalin or formaldehyde. Then the histological sections, the tissue is going to be sliced into thin section, just 1 or 2 cells thick. And then we're going to have to stain it. And so the tissues, a lot of the structures or the biological molecules are clear, and so you won't really be able to see them, so you need to use special tools. And these stains, and these special stains tend to have an affinity, or they will tend to adhere to some structures more than others. So this allows us to stain different things depending on the structure and what kinds of dyes or stains like those particular structures. So those stains will bind to those different cellular components. And then sectioning. Sectioning is going to reduce the 3-dimensional structure to a 2-dimensional slice. So that's what I was talking about earlier. When you section it, when you cut it into sections, you're cutting a three-dimensional thing, you're looking at it in 2 dimensions. There'll be some examples here in a bit to kind of visualize what we're talking about. So here we have something we are familiar with. This is let's say this is a hard-boiled egg here in the middle, right here. And if we cut it along different locations, right here, we're going to see different things. Along this dotted line here, if we look at it from the cut and we look at it from the inside of the cut, we look at that section, it's going to look like this. You're not even going to get any yolk in there. And then if we cut it a little bit further inward, then we're going to start to see the yolk, but we didn't cut right through the middle of it. And then if we cut right down the middle, we'll be able to see the yolk, but the yolk is, we'll see it much bigger. And the same on the other side. So we see what is occurring on the other side. So when we talk about actual tissues, the cell's going to have essentially a located nucleus. And so some of the slices that we cut through, when we cut through the tissue, we're going to miss the nucleus. And then some slices, the nucleus is going to appear smaller, just like in the analogy with the hard-boiled egg over here on the left. And so what about tubular structures, like ducts and so on? When we cut through those tissues that have these tube-like structures, and what would we get? So here, right here, we have an example. It looks like an elbow of a pipe or so. Well, let's say this is a curving of a microscopic tubule. And if we cut it through here, right here, and then we look at it from the cut here, from the section, that's going to just look like 2 separate circles. And then if we cut it a little bit lower, right here, then it would look like this here, where you get actually a part of the rest of that tube towards the bottom. And then if we cut it even lower, then this is all we're going to see. So you see, depending on where it was sectioned, you're going to look at different deals here. So here we see this again here with a long wavy tube, maybe spiraling downward into some tissue here, some kind of duct that maybe goes to the outside, maybe like a sweat gland or something like that. And if we were to section it, we're going to get areas where we section through it longitudinally, but because maybe it's spiraling inwards, in some places we're going to get oblique sections of the tube and even complete cross sections where you have this perfect circle-looking structure there. So these three-dimensional structures will look different depending on where you section it from. So what kind of sections can we have of tissues or cells? And we use a bone here as an example. We're going to cut through it here. We cut through the bone lengthwise, and that would be called the longitudinal section, and then you'll see it on slides as LS. So let's say we have a muscle tissue. Muscles tend to have real long fibers, or their cells are called fibers. So if we cut longitudinally through it, then you will see a long fiber. But if you do a cross section, like the way they're doing through this bone right here, and you were to cut through that muscle fiber, then you're just going to get a little circular looking structure for the cell. And then the abbreviation for cross section is CS or XS, or sometimes TS, because it's also called a transverse section, just like we studied in the lab. This cross section would be cut perpendicular to the long axis. And then we have an oblique section here. So this is what it would be like if we cut at an angle through there. We get more of an oval shape if the structure was cylindrical in nature. So the oblique section is going to be cut at an angle somewhere between cross section and longitudinal section. Then we have smears. So a smear then is when a tissue is just rubbed across a slide. For example, blood's a liquid, so we just rub it across a slide, and then we look at the slide under the microscope. That would be called a smear. And then we have a spread. A spread is like when we get some kind of a membrane that's fibrous and cobwebby, and we just lay it down on the slide. An example would be areolar tissue. And so that would be called a spread.